Good afternoon, class. We want to start a lecture on the hypersensitivity reactions. And before we start this lecture, we want to watch a video. In this video, I want to give, with this video we are about to watch, I would like to give credence or acknowledgement to the owner of the video, that is Frank Lectures. They did a summary of the 4 types of hypersensitivity reactions, and they did a summary of it, and it's so wonderful. So before I teach my students, I would like to acknowledge them. That's Frank Lecture. That anybody, any students all over the world, you want to watch any or have a lecture on or have an overview of hypersensitivity reactions, you have to go and subscribe to the Frank's Lecture in order to gain an in-depth understanding into the mechanisms of what hypersensitivity reactions. So let's now zero in on the video.
At this point in our lecture series, we understand how our immune system provides protection against pathogens, diseases, and infections. Our immune system is able to recognize and distinguish between self and non-self antigens. By using a wide variety of defense mechanisms, the immune system eliminates potentially harmful antigens from the body. But sometimes, the defense mechanisms of our body can cause damage, instead of providing protection. At this point in our lecture series, we understand how our immune system provides protection against pathogens, diseases, and infections. Our immune system is able to recognize and distinguish between self and non-self antigens. By using a wide variety of defense mechanisms, the immune system eliminates potentially harmful antigens from the body. But sometimes, the defense mechanisms of our body can cause damage, instead of providing protection. This happens in 2 situations. First, when the immune system overreacts to certain foreign antigens. Second, when mechanisms of self-tolerance fail. Now look at the term hypersensitivity. Hyper is derived from Greek term hyper, which means over, beyond, or above. And sensitivity means the ability to detect and respond to slight changes or signals. Hypersensitivity is defined as any immune response that is excessively above normal. It is an inappropriate immune response that results in host damage. hypersensitivity reactions are the consequence of adaptive immune responses that result in tissue damage. These reactions are manifested when the individual gets second or subsequent exposure to the antigen. Hypersensitivity reactions can be grouped into 4 types. This classification is based on the effector mechanism involved, or in other words, the components of immune response involved in the hypersensitivity reaction, and the time of onset. That is, how quickly the reaction develops. In the coming video lectures, we will study each of these hypersensitivity reactions in detail. For now, let's have a short introduction to each of these hypersensitivity reactions. Type 1 hypersensitivity. Type 1 hypersensitivity reactions are IgE-mediated reactions. In this case, antigen-specific IgE antibodies bound themselves to cells such as mast cells and basophils. When these antibodies interact with the antigen, degranulation of cells occurs. This degranulation releases inflammatory mediators which further result in symptoms of the reaction. Type 1 hypersensitivity is also known as immediate hypersensitivity since it occurs immediately upon exposure to the antigen. It takes only 2 to 30 minutes to develop. Examples of type 1 hypersensitivity reactions include allergies to food, pollen, insect stings, etc. Type 2 hypersensitivity. Type 2 hypersensitivity reactions result when IgG or IgM antibodies recognize and react with antigens present on cells or tissues. These further result in activation of complement and phagocytosis. The cellular destruction takes place that ultimately result in inflamed and damaged tissue. Examples of such reactions include blood transfusion reactions, hemolytic disease of newborn, and drug-induced hypersensitivity. Type 2 hypersensitivity reactions are also known as antibody-mediated cytotoxic hypersensitivity. The term cytotoxic tells that these reactions result in destruction of host cells. Type 2 hypersensitivity reactions take 5 to 8 hours to develop. Type 3 hypersensitivity. Type 3 hypersensitivity reactions are caused by immune complexes or antigen-antibody complexes. These insoluble immune complexes are formed by antigen and specific IgG antibodies. In the normal situations, small and soluble immune complexes are phagocytosed by dendritic cells and macrophages. But insoluble immune complexes deposit at various sites in the body. For example, in the walls of blood vessels. Their accumulation results in the activation of complement proteins. The resulting inflammatory response damages the tissue. Arthritis and glomerulonephritis are the consequences of type 3 hypersensitivity reactions. Type 3 hypersensitivity reactions are also known as immune complex-mediated hypersensitivity. It takes 2 to 8 hours to develop. Type 4 hypersensitivity. Type 4 hypersensitivity reactions are mediated by antigen-specific effector D cells. This is the only class of hypersensitivity reactions which are triggered by T cells and therefore also known as cell-mediated hypersensitivity. Examples of such hypersensitive reactions include nickel allergy and allergic reaction to poison ivy. Type 4 reactions are also called delayed type hypersensitivity because they become apparent 1 to 3 days after exposure to the antigen. So, this was a short introduction to the hypersensitivity reactions. In the next video lecture we will study in detail type 1 hypersensitivity and its underlying mechanisms. If these video lectures are helpful to you, please like, share and comment. Also, do subscribe if you are new to this channel. Thank you for watching.
So back to our lecture. We would like to thank Frank Lecture for giving us this in-depth knowledge and also exposition into hypersensitivity reactions. So Frank Lectures, we are grateful to you for your acumen in immunology, especially in hypersensitivity reactions. So if you want other topics on immunology, just go to the link of Frank Lectures, click on the link, like their link and share. Thank you very much, Frank Lectures. So back to my students. Okay, so hypersensitivity is what? It refers to undesirable immune reactions produced by the normal immune system. And this is also an exaggerated or inappropriate reactions harmful to the host. Also, hypersensitivity or allergic reactions are made up of 4 different categories based on its mechanism and also the time taken for the reaction. A disease condition may involve more than one type of the reaction. For instance, if you have autoimmune diseases, that's failure of self-tolerance and subsequent immune response against itself. So let's see. For instance, if I'm exposed to dust, what happens to me is that if I have an allergic tendency, then I will end up getting congestions. I'll end up getting nasal congestions, bronchial constrictions, and the rest. Pulmonary diseases, just because I inhaled dust particle or pollen grains. So there are some of the times that your own antibodies will tend to fight against your own cells. Your own antibodies tend to fight against your own cells. So in such situations like that, we have, for instance, we would like to know why in diabetes type 1 there is an autoimmune destruction of the beta cells of the islet of Langerhans. Of the islet of Langerhans, that's pancreatic cells. There is autoimmune. The body will produce antibodies against the pancreatic islets or the beta cells of the islet of Langerhans, and this will lead to diabetes type 1 or insulin insulin dependent diabetes mellitus. This will lead to insulin dependent diabetes mellitus. So what's the cause? You are there and you just realize that your antibodies and cytotoxic cells are produced and they start attacking your pancreas. These are all autoimmune. Your own body cells fight against your own defensive mechanism or antibodies or T cells, cytokines fighting against your own cells, and that is not what is supposed to happen, because they are rather supposed to fight against parasitic infections and foreign agents. So this is what happens. This autoimmune response or autoimmune disease. This leads to what? Failure of cell tolerance, because your antibodies are supposed to tolerate your antigens, which are your endogenous antigens. That the antigens belonging to your body are called, or antigens located or produced in the body are called endogenous, and those coming out of those coming from outside, antigens coming from outside are called exogenous, and those produced inside the body are called endogenous. Those coming outside the body are called exogenous antigens. So with autoimmune responses, there is what? Attack or onslaught against antigens that are located inside the body, and these are what? Endogenous antigens. And these ones can be as a result of molecular dysregulation, mutations in the DNA, and other inborn errors. So let's tackle this in depth. Okay, so we have type 1, which is immediate hypersensitivity. If you inhale pollen grains or perfume, immediately you start to get nasal congestion or what is called catarrh. Your bronchioles become constricted and you find difficult to breathe. You have what? Chest pains, and the type 1 is mediated by immunoglobulin E. That's IgE. The IgE regulates type 1 hypersensitivity reactions, and what it does is that it binds onto mast cells. So if this is a mast cell, this is the nucleus of the mast cell, this is the cytoplasm. So IgE will then bind onto the mast cell. Then after binding to mast cell, it will bind to antigens. So let's see. So it will bind onto mast cell like this. Using this Fc fragment, it will bind to mast cell. Then use this fragment antigen binding. Then that one, it will bind onto what? It will use this one to bind onto the antigen. This is the antigen. This is the antigen. So when something like this happens, there is degranulation. So when something like this happens, then the mast cell will now release or degranulate or release its cytoplasm. So upon releasing its cytoplasm, it contains vasoactive substances which cause vasodilation. Thus it's producing histamine, which we call vasodilation, bronchial constriction, and a whole lot of dangerous reactions are going to occur in the body. And if this reaction is, if the antibodies are circulating, then it will lead to anaphylactic shock or systemic shock. But if this antibody anaphylactic shock, because somebody can just inhale perfume spray and the person will collapse. That is an anaphylactic shock because the person will start producing high levels of histamine. So the medicine or the drug to control high level of histamine production is what? Antihistamine. Antihistamine. So the person has to be given antihistamine. And also in excessive case, when there is systemic reaction, that is the reaction occurring in the blood because of the antibodies, circulating antibodies in the blood, then you need to give epinephrine or norepinephrine to be able to arrest that, to be able to arrest the hypersensitivity reaction. So one may ask, how can you be able to remedy the hypersensitivity reaction, or how can you be able to stop the reaction? So norepinephrine can also be given in systemic anaphylactic shock. Epinephrine, systemic anaphylactic shock. Then, for you to be able to arrest the condition, you need to abstain. You need to abstain from anything that makes you to react, whether it is food that you took in that gave you rashes, urticaria, hives, or whether it is medicine you took that made you vomit excessively, or defecate, or lose your bowels, have diarrhea, then you have to stop those medications. So anything that triggers or anything that sensitizes you and triggers a secondary response to a non-disease causing pathogen, you need to remove or abstain from it. So in hypersensitivity reactions, the antigen over here is not it's not a pathogen. Antigen is not a pathogen. In hypersensitivity reaction, the antigen is not a pathogen, but rather it is what? It can be perfume spray particles, perfume spray particles. It can also be pollen grains, drugs, that is drug idiosyncrasies, drugs, food, as in food allergies. Some foods, when you take them in, you will get a lot of allergic reactions. So foods which trigger allergic reactions, food allergies, and so on, etc. So let's proceed. So we have the type 2, that is antibody mediated, type 3 immune complex mediated, and type 4 T cell mediated. So let's see. So the reactions we use to be able to quantify the amount of antibodies binding to cells, especially the red blood cells, is Coombs test. In Coombs test, we have the indirect and the direct Coombs test. We'll be doing it in hematology. Okay, so type 1, commonly called allergy or allergic reactions, mediated by immunoglobulin E antibodies produced by plasma cells in response to stimulation of T helper 2 cells by an antigen. The antigens that stimulate are called allergens. Examples are house dust or household dust, pollens, cosmetics, insects, clothing, and drugs. Exposure may be ingested. Inhalation. Exposure may be when you ingest it or eat it, when you inhale it, when you inject it into your veins, or when you inject into your muscles, intramuscularly, or direct contact. Type 1 hypersensitivity reactions can be systemic. Example, systemic anaphylaxis, or localized to a specific target tissue or organ. So when the reaction occurs because the immunoglobulin E is floating in the blood and forms complexes with the pollen grains in the blood, then we say it is a systemic anaphylaxis, and this is very life-threatening. But if it is localized to an organ or a particular tissue, then we say it can be what? Asthma, because asthma is localized to the respiratory system. Then allergic rhinitis is localized also to the respiratory system, especially the nasal sinuses, nasal cavity. So type 2 hypersensitivity involves immunoglobulin. Okay, so we are on type 2 hypersensitivity reactions, and this one involves immunoglobulin G or immunoglobulin M, antibody mediated. So it's an antibody mediated cytotoxicity. So we have immunoglobulin M or immunoglobulin G triggering the reaction, and this one the immunoglobulin M, immunoglobulin G, immunoglobulins react with cell surface antigens to activate the complement system and produce direct damage of cell surface. Examples are found in transfusion reactions and hemolytic disease of the newborn. So let's see, how does transfusion reaction occur due to type 2 hypersensitivity reactions? So let's see. So if you have blood group A, if you are blood group A positive, then this is what a donor. The recipient, if the recipient is blood group B negative, or let's say, okay, let's see, so blood group B positive is here, so A negative. Okay, so let me put it well. So we have, let's see, so let's get the colors. This is one blood group, another, don't mind my drawing, another, okay. So if this is A negative, A negative means that it's the only antigen it has is what? A. It has no RH. Then let's say the A is what? Like this. B, this is B negative. It has antigen B on its surface. This is O negative. It has no antigen whatsoever on its surface. This is A positive. A positive is going to have what? A as an antigen, Like the first one, A is an antigen here. Let me clean it and write it well. A is an antigen here. Okay. A positive. Okay. So this is A positive. A is an antigen here. Then the RH is also here. So RH, that's also an antigen. Then you go to B positive. B positive has what? B as an antigen. Then RH also here. Then O positive. O positive has no AB group but has only the RH. And this A antigen, B A antigen, B antigen, they are constituted by what? They are formed by glycoproteins on the surface of the red blood cell. So the red blood cell surface or their plasma membrane contains glycoproteins. An example is what? Mannose. Another one is called fucus. Another one is called N-acetyl glucosamine. Glucose. N-acetyl glucose amine. Another one is called N-acetyl galactosamine. Amen. Okay, so these are the four. They are more than that, but they are about 5, but I'll put 4 here. These are what? Glycoproteins on the surface of what? Red blood cells, and that is what gives them their antigenic determinants. That makes one blood group different from another. Antigenic determinants. That is what gives them the different antigen epitopes. Antigen is the same as what? Antigen is the same as epitope. Okay, thank you. So with this, A negative will produce antibodies against what? The B. And B will also produce antibodies against the A, because A negative cannot produce antibodies against A. If it produces antibodies against A, that is what we call autoimmune response or autoimmune reactions. It can't produce autoantibodies. It has to produce antibodies against another cell. If it produces antibodies against itself, that means it's going to kill itself. So this is the principle behind blood transfusion. The A will produce antibodies against B, and the positive will also produce antibodies. Negative will produce antibodies against the positive, the RH positive. So this is the principle. We'll go much more into detail when we get to second year and we are doing immunohematology or blood transfusion reactions. So transfusion reactions are hemolytic disease of the newborn. Examples of type 2 hypersensitivity reactions. Type 3 hypersensitivity is also known as immune complex hypersensitivity. The reaction may be general that's serum sickness, or may involve individual organs including other organs. Antigens causing immune complex mediated injury are exogenous or endogenous. So immune complexes. So we have, so if this is what, if this is, so immune complexes, most of the time they circulate in the cell. We have circulating antibodies in the cell, in the blood vessels, circulating antibodies in the blood vessels. So this is a typical antibody. So antibody, antibody, light chain is gone. So they form immune complexes. So we have another antibody, another one, another one, another one. We have another one, another one. Then the third one is here. I'm just drawing immune complex. How is it is. So this is it. So there is what antibodies over here. There are antigens over here. Antigens over here. Antigens. So this one forms an immune complex. And this immune complex, this big thing, is circulating in the blood. And they get dislodged. They get dislodged into joints. They get dislodged into capillaries. They get dislodged into kidney and other places. Glomerulus of the kidney, where there is ultrafiltration of blood to produce urine. So this is an immune complex. This whole thing is an immune complex. So this immune complex will attract complement. It will attract complement system proteins to come and cause a reaction. It will also attract cytokines to the site to cause a reaction. Cytokines and lymphokines to come and cause what? An immune reaction, which is devastating. And this is as a result of what? This is what leads to rheumatoid arthritis. This leads to rheumatoid arthritis, glomerular. It also leads to glomerular nephritis. That's glomerulus of the nephritis, glomerulus of the kidney. Nephritis, inflammation of the kidney, of the glomerulus of the kidney, glomerular nephritis. So let's continue. So the reaction may be general. Okay, antigens causing immune complex mediated injury can be exogenous, that's outside, coming from outside the body, or already inside the body. If it is already inside the body, this one will trigger what? Or it is called autoimmune reaction, because the body is raising antibodies or cell-mediated response or chemicals to fight its own cells. That is what we call autoimmune response. So antigens combines with antibody within circulation and form immune complex like I demonstrated. Complexes are deposited in the body and activate complement system. Polymononuclear cells like the neutrophils are also attracted to the site. And this results in inflammation and tissue injury. Diseases in which you can find immune complex deposited are what? Hypersensitivity pneumonitis, that's inflammation of what? The lungs and respiratory tract. Then we have the glomerulonephritis, rheumatoid arthritis, systemic lupus erythematosus, that's lupus. So all these are, so systemic lupus erythematosus, autoantibodies will be raised against the body's antigens like the DNA and the rest. and there will be reaction. So inhalation of antigens into lungs stimulates antibody production. It's true. Subsequent inhalation of the same antigen results in formation of immune complexes, which activates complement system. So in type 3, complement system is activated. Glomerulonephritis is an immune complex in the blood, or it occurs when immune complexes are deposited in the blood. It occurs when immune complexes in the blood are deposited in the glomerulus. So the glomerulus is part of the nephron of the kidney. So if you have the nephron of the kidney, so let's see. So if this is the afferent artery, this is the glomerulus, the Bowman's capsule is here. Bowman's capsule. Bowman's capsule surrounded Bowman's capsule. We also have the efferent arteriole, which contains the and the afferent arteriole, which contains the juxtaglomerular apparatus. Then we have the distal convoluted tubule, which comes off here, which contains the macula densa, and this one contains the podocytes and the rest for ultrafiltration into the proximal convoluted tubule. And the proximal convoluted tubule leads to what? The nephron, or the loop of Henle, and the loop of Henle goes to the ascending tube of that, which also goes there and ends in the distal convoluted tubule, which also ends in the what? Collecting duct. And the collecting duct concentrates the urine, and we know the urine is formed through the urethra. So this is a sketch of the glomerulus. So if there is immune complex inside this, then you know that blood cannot be filtered into the proximal convoluted tubule to form urine. And this one will lead to kidney failure and ultimate death results. Okay, rheumatoid arthritis is when immune complexes are deposited in the joint, and which results in release of what? Inflammatory chemicals. The joints begin to break down and become distorted. This trigger is not well understood and is treated with anti-inflammatory drugs. So an example of an anti-inflammatory drug is what? Ibuprofen. Aspirin, and the rest. So they are under a class of drugs called NSAIDs. And if you have ulcer, you shouldn't take it because it's acidic. So these are the anti-inflammatory drugs you take when you are having what? When you are having rheumatoid arthritis, so that you limit the inflammation, reduce the inflammation. Systemic lupus erythematosus is also under type 3. When it occurs when autoantibodies or antibodies of the body. It occurs when autoantibodies are produced against the DNA of the host or of the same person, resulting in immune complex formation. So autoantibodies see the DNA of the person or of the host as what? As foreign, or as a disease-causing organism, and the antibodies are released against the DNA of the same person, and this is an autoimmune disease. Many other autoantibodies can also occur against red blood cells. Autoantibodies produced against platelets, lymphocytes, muscle cells. And this one, the trigger is unknown, what causes it is unknown, and you have to take immunosuppressive drugs to reduce autoantibody formation. Glucocorticoids can also reduce the inflammation. It's a type of what? Glucocorticoid is a type of what? Steroid hormones that we can use to reduce stress and also reduce inflammation. Okay, so we are on type 4. That is the delayed or cell-mediated hypersensitivity. This takes a long time to occur. When one is exposed to the antigen, it takes a long time for the reaction to occur. The first stage is a sensitization stage, and the second stage is the effector or the secondary response stage. Delayed hypersensitivity is a function of T lymphocytes and not antibody. So here antibodies do not mediate it. The first 3 types of hypersensitivity reactions are mediated by antibodies, but this type 4 is mediated by T lymphocytes. It starts hours or days after contact with the antigen and often lasts for days. That's why we say it is the delayed type or cell-mediated delayed type hypersensitivity reaction. It can be transferred by immunologically committed or sensitized T cells, not by serum. Principal pattern of immunological response to variety of intracellular microbiologic agents, that is Mycobacterium tuberculosis, viruses, fungi, and parasites. So, principal pattern of immunological response to variety of intracellular pathogens, such as, so most of the time this one happens in what? Nucleated cells. This type, it attacks nucleated cells, and it uses the MAC class 2 molecules. It's mediated by the MAC class 2 molecules. So it presents the MAC class 2 molecules presents antigens to helper T cells, which have CD4 receptors. I explained this one in the previous lecture on MHC. So let me see whether I can recap. So for the cell-mediated response or cell-mediated cytotoxicity, this is what we have. So we have okay, so so if this is an antigen, from outside or endogenous source, maybe this is a malaria parasite, it will have what? Antigens on its surface. So it has what? Antigens on its surface. So what happens here is that if this is an antigen presenting cell, that's an APC. And examples of antigen presenting cells are the dendritic cell found in tissues or the skin, dendritic cell, we also have the B lymphocytes, macrophage. So they are antigen presenting cells. They will swallow the antigen and present it on an MHC peptide on the plasma membrane. So if this is an MHC peptide, so the cell is an antigen presenting cell. So after So let's say it is a macrophage or a dendritic cell. So let's name it as what? Either a macrophage. I like the macrophage. So the cell is a macrophage. Macrophage. It's a macrophage. So macrophage. Then it swallowed the peptides. It swallowed these peptides. So these red peptides, these antigens. Then put them into vacuoles. Then presented them on its surface like this. And it can present this antigen for destruction to T helper cells. So if this is a T helper cell, it has receptors for the antigen. So T helper cell receptors for the antigen. Let's make it black. So these are its receptors for this antigen. These receptors. So T helper cell receptors. So the receptor will come and bond onto. So there will be a linkage. They will come together. There will be a linkage. So that this one will now release, the T helper cell will now release what? Cytokines to destroy interleukins, interferons, tumor necrosis factor, transforming growth factor, and the rest, to destroy these antigens. And after destroying the antigens, they are removed from circulation. And so this is what so this is T helper cells, and this is what? Macrophage. Sorry for the spelling, it's supposed to be MA. Macrophage. Thank you. So let's go to our explanation. Okay, so principal pattern of immunologic response to variety of. So I forgot this. So these molecules, these blue colored molecules are there. This one is the MHC class 2. MHC class 2. If it is in human beings, it will be what? HLA 2 molecules. They present MHC class 2 molecules present antigens to what? MHC class 2 molecules present antigens to what? T helper cells. Principal pattern of immunologic response to variety of intracellular microbiologic agents, that is Mycobacterium tuberculosis, viruses, fungi. So this is what happens. Okay, so clinical importance of delayed hypersensitivity reactions. So this is found in what? The tuberculin response. That is, if somebody wants to travel outside or you want to test somebody whether he has tuberculosis, an injection of tuberculin beneath the skin causes reaction in individual exposed to tuberculosis or tuberculosis vaccine, and it's used to diagnose contact with the antigens of Mycobacterium tuberculosis. So if after injection of the tuberculin under the skin, if there is no response, then the individual will not be infected or having been exposed to a vaccine. But red or hard swelling develops in individuals previously infected or immunized. So this is an example. This is the swelling. So this means that the person has been immunized before or has gotten the infection. Graft rejection. This is very important. Rejection of tissues or organs that have been transplanted. For instance, if somebody is having a kidney failure and you go to South Africa, go and buy organ from an organ bank, and you bring it to the person's body will reject it because it's totally different. The other kidney has different major histocompatibility complexes which are totally different from the recipients. So it will become difficult. But you can easily transplant organs between Siamese twins or even identical twins because they look identical. Their genetic makeup is identical. So with them, you do not need to give them too much of immunosuppressant drugs. Grafts perceived as foreign by a recipient undergo rejection. A graft is what? Any part of the body you want to substitute in somebody who has an organ failure. So if you borrow an organ, you buy an organ from an organ bank, come and put it in somebody who has an organ failure, we call it what? Graft. And if there is the person's body says, no, I don't know this organ is totally different from my genome or my genes or my genetic makeup, then the body will reject it to produce antibodies against the organ, and there will be autoimmune response. So immune response against foreign MAC on graft cells, very important. So if you have a graft cell or an organ coming from an organ bank, it will have what? Foreign major histocompatibility receptors on its surface. So the host, because it's foreign to it, it will reject. The host immune system will reject it by producing antibodies against it. Rejection depends on degree to which the graft is foreign to the recipient based on the type of graft also. It's important. So let's see. For instance, autograft is like this. This is a mouse. You remove the mouse, you remove tissue here and put it around the chest. We call it autograft. There will not be any rejection. It's like this. We have something that somebody has gone to do, a celebrity has gone to do. We call it liposuction. Liposuction. Removing tissue from one place and putting it in another place. And we also have plastic surgery. Plastic surgery. You remove a tissue from one part of maybe your buttocks. You remove a tissue from your buttocks and put it in your thighs. Or you remove tissues from your belly, your abdomen, and put it at your buttocks so that you have more buttocks. Or you remove tissues from your buttocks and put it in your breast so that your breast will be bumpy or plumpy. That is all what we call plastic surgery. And there will not be any tissue rejection, or your antibodies or T cells will not be produced against the tissue you are conveying from your buttocks to your breast, because it's the same body, the same genetic makeup of the same person. But imagine you were doing it, you were transferring these tissues from your husband's abdomen into your buttocks. There will be rejection because your husband is totally different from you. So if it's an autograft from one's own body to his own body, no rejection, or the rejection is very minimal. If it is between identical twins, genetically identical Sydney or clone, it is possible. Rejection is very minimal. But if it is between you and your sister, or you and your husband, you belong to the same species as Homo sapiens, but you have different genetic makeup, so it's not possible. Genetically different member of the same species, so it's difficult. Then if it's another species, like you go and take a liver from the cow and come and put in somebody who has a liver failure, a human being who has a liver failure, there will be great organ rejection, tissue rejection, because it belongs to another species. Their major histocompatibility complex is genetically different from what pertains in humans, so there will be total graft rejection. Ladies and gentlemen, look through this mechanism and study it vividly. This brings us to the end of our class. All the best in tomorrow's mission. Thank you very much.